Alrighty, is this working? Yes. So a little bit of organised chaos. We're going to rearrange the program a little bit. Treena Burgess from Murdoch University is sadly unwell today, so Giles Hardy is going to be presenting in her place on molecular tools for the detection of Phytophthora pros and cons. Take it away. Here's your... Sorry to throw you in the deep end, Giles.
That's all right. I wasn't expecting that. And especially I didn't expect Treena to lob me in at the last minute. So we've been working on detecting Phytophthora for a number of years, and there are lots of issues associated with diagnostics and detecting Phytophthora, and we use both molecular and the traditional tools. And the traditional methods for detecting Phytophthora include direct plating of plant tissues onto selective agar media, or indirect baiting, as you can see with the leaves floating on the water in those dishes where zoospores are produced out of roots and soil. They infect the leaves, then we isolate out onto selective agar. And the former method is much more common for foliar Phytophthora species and agricultural species, and is usually really quite robust. And the latter, where we use the indirect baiting from soil with what we're seeing here on the right of the image, is used in the environmental samples, and it's not always foolproof. And we've been doing a lot of work, both ourselves and other labs, at actually optimizing these methods to make them more efficiently. And so I'm going to talk a little bit about these now. And one of the things we're finding is that we're becoming more and more aware there are multiple Phytophthora species present in soils, and we're trying to work out ways of getting all of these species out in the baiting procedure and understanding why sometimes we know a particular pathogen is present, we don't get it. And this PhD student, Suchana Saka, has been showing that in soil samples with mixed species, that those that produce sporangia and release the motile zoospores faster will preferentially colonize the baits and can mask other Phytophoras. So in the top diagram, there you can see with the closed black boxes is Phytophthora cinnamomi, and it's got very low number of lesions on baits compared to Pseudocryptica, which is the open circles above it. And so it's masking Phytophthora cinnamomi, and we don't isolate it as readily as cinnamomi as readily as we'd expect. And that can vary between species. There's just 3 examples here. And she's done this sort of work with about 35 different Phytophthora species that we isolate quite frequently. And so one of the benefits of soil baiting is that we can use large sample sizes, which is very different to the molecular methods. It's a very simple technology, and you can get true positives. You can get an isolate into culture, which you can then do pathogenicity testing on and explore its ecology more, etc. It is time consuming. You can have isolation bias. You could miss things because maybe you're not using the right baits. And it's not that sensitive always. And you can get false negatives, as for the example in the top diagram where we know cinnamomi is there, but it's been masked by Pseudocryptica. So you come with a result saying there's no cinnamomi in that soil sample, when in fact there is. So then we have the molecular techniques, and these have been developed also for the detection of Phytophthora. And you have 2 methods, the PCR, the polymerase chain reaction, which can be based either directly from plant tissues, or when we've isolated the pathogen onto agar, we can extract DNA and then determine what you've got. Or the metabarcoding methodologies, which use species-specific or genus-specific primers. And the idea here is you can identify multiple species from across taxa in a single soil sample, or you can just be home in on Phytophthora species in general. So if we explore these a little bit more, the primer-based PCR assays are most commonly used, and at the moment, and much more emphasis is put on the qualitative PCR, the qPCR, and the sensitivity of these methods can be improved by getting very specific probes, or designing very specific probes to target particular Phytophthora species, which I will come back to in a moment. And in the literature, there are numerous primer pairs that have been developed for particular phytophoras, and in this case, Phytophthora cinnamomai. And they say they're species specific, but when you go and test them, you find that many of these primer pairs that have been developed and published are not that specific. And one of the reasons, so you get a lot of false positives. And one of the reasons why they're not specific is that the people who've designed these primers have not tested them against a lot of other different Phytophthora species, especially those, for example, that are in the same clade as Phytophthora cinnamomai. So those technologies need to be improved a bit. So this is what I'm showing here, using the PCR-based molecular methods, was work by Manisha, another PhD student of ours, who was involved in an eradication project where we wanted to confirm that we'd actually got rid of Phytophthora. We'd killed Phytophthora during the eradication process, and we wanted to confirm that Phytophthora wasn't present and use molecular and baiting techniques together. But we know that DNA can survive in soil for quite a long time. So she looked at putting Phytophthora cinnamomai DNA into 5 different soil types, and either in wet soil or dry soil. And you can see from the slide here that in the dry soil, which is the closed triangles, the upper line, the pathogen can survive 60 days, 200 days, and more. In wetter soils, the DNA tends to disappear more quickly. So if you're looking at using DNA as a diagnostic, it's not going to tell you whether the pathogen is still alive or not. It's a very fast procedure. It's also very sensitive. But unlike those big containers where we can bait kilograms of soil at a time, you can only do milligrams when you're extracting from soil for molecular purposes. So that's the limitation. You know, small sample size and sampling strategy. How many samples do you collect from a site to confirm that Phytophthora is not there, for example? We don't know whether the organism was alive. It is also expensive. So Manisha also then wanted to, we still wanted to determine, okay, have we killed the pathogen or not? So we looked at RNA, and we know that RNA only survives for a maximum of a few days after an organism dies. So we looked at extracting RNA, converting it to complementary DNA, and then conducting the same assays in the soils that we looked at in the previous slide. And she showed that it disappeared within hours to a maximum of 3 days. So by using RNA as a diagnostic, you could show very quickly whether your eradication methods had worked or not. However, while it was very specific, she was able to show that it was no more sensitive than baiting. And cinnamomai needed to be actively growing in order to detect it using the RNA. So let's think about this a little bit more. In a gravel soil, or in a haul road, or in other sites, the pathogen isn't growing. It's not in plant tissue. It's not in living plant tissues. It's dormant. It'll be in roots, and it'll be dormant, so it's quiescent. So it's not producing any messenger RNA, so we can't pick it up. So in order to pick it up, we need to make it germinate, where we put it into water and use baits. And hence, that's why I say it's really no more sensitive than baiting using this RNA technique, even though it's very sensitive. So what we now suggest is that we use a hybrid method, which uses a combination of the RNA and the baiting. And using this method, we can pick up very, very low levels of Phytophthora cinnamomai. So we can improve the diagnostic methodology using this hybrid method. And coming back to the metabar coding, where we can look at a range of taxa from a particular sample, which we call eDNA. And this has become very common, and a lot of people started to do that. And we're one of the first labs to have done this with Phytophthora anywhere, and we've developed Phytophthora-specific primers. Originally, we started out using primers that had been developed by other people, and on the right-hand figure, on the right-hand side of that figure, there's an example of DNA from 80 different Phytophthora species put into soil, including cinnamomai, and we were targeting cinnamomai. And using those primers developed overseas, we found there's a very poor relationship between the DNA concentration of the PC and the proportion of reads. So we had to go and develop our own primers, and we actually ended up getting very specific primers, and that's the one on the left diagram there, where we got a very good relationship of 0.65 in terms of the DNA concentration of the proportion of reads. So again, the metabarcoding is a useful tool, and we can and it can assist us in diagnostics. But you need to use primers that are specific, and you need to test them comprehensively against many different Phytophthora species, particularly those that are related to the Phytophthora species of interest, like Cinnamomi, so in the same clade as Cinnamomi. We then had another student who looked at 5 different soils and isolating Phytophthora species from these soils. And in the table, you can see there are 30 different Phytophthora species he isolated. And he used the traditional techniques by baiting, filters, or putting soil or roots into an apple. And then he used the metabarcoding directly from soil, from filters, from field roots, and glasshouse bait roots, where we collected soils and germinated a range of susceptible seedlings into that soil. Once they germinated, we'd take the roots out and then extract the DNA. And you can see vast difference between the isolations using traditional baiting. We only got 6 Phytophthora species out of those soils. So when you see the number 2, it means that say looking at the multivora line, Phytophthora multivora, we've got 2, 1, 5, 5, 3, 5. The 5 means 5 soils. You've got Phytophthora multivora out of 5 soils, or 1 means 1, depending on whether it's the metabarcoding or multivora. But you can see or the direct isolations. And you can see here huge differences too with the metabarcoding between just collecting from soil, 13 species, filters, 24 species, field roots, 25, and then the glasshouse bait roots, 19 different species. So it's quite accurate, and there are a lot of other Phytophthora present. And this is one of the reasons we came back and did the work, which I showed in the first slide, where we're looking at the different, the speed at which different Phytophoras produce the sporangia and release the zoospores, because we're aware we're getting all these Phytophoras, they're present in the soil, but why aren't we isolating them? But something useful that came out of this was that when you're providing baits to diagnostic labs, get more roots than soil. Put the emphasis on collecting roots, because you're going to get many more hits. Your success rate will be much more successful using roots than it will just about soil. So keep that in mind. So the main advantage of metabarcoding is it's very, very sensitive, especially if you're targeting specific taxa. And the results, as you can see in the previous slide, were very useful in terms of ecological studies, determining what's there, what's present, and providing you a good baseline. And if you do find an unexpected species or something that shouldn't be there, or a species that has never been described in Australia before, say something like Ramorum, Phytophthora ramorum, which we don't want in Australia, then you could concentrate and do your baiting. You could go back to the soils and try and isolate it out into culture. So it's extremely sensitive method. It is expensive, and it is quite time consuming. There is still some inability to detect some species, especially those that are very closely related. So you can get, if you like, species complexes with some of the primers. And so you can also get false positives. And depending on how clean your laboratory is or how dirty your laboratory is, you can get contamination issues. So you can get false positives as a result of DNA from previous studies being done in that lab. So hygiene is very important. And again, you don't know if the organism is alive or not using these methods. So thank you. No questions, that's good. Are you allowed to ask questions?
Yeah, sure we
Just
so on this, one of the slides here, so one of the criticisms of metabar coding is that it's not quantitative. Can you explain that a little further? Why that?
You can't. It's not going to tell you how much Phytophthora is present. So what the inoculum potential is, if you like. Yeah. So if you've got one chlamydospore, thousands of chlamydospores. So
isn't the greatest strength just in the qualitative assessment?
Yeah, well, that's right. It's still really good. But sometimes, again, like with the eradication, with the baiting, we go out, we show, you know, when we started, we'd get 500 isolates. We come back 6 months later or 2 years later, we only get one recovery. So obviously the inocular potential, the amount of Phytophthora present inside is declining. It'd be nice to be able to sort of show similar. But yeah, plus or minus is really important. Really, we want minus. Don't we? Yeah.
Okay.
Emo, you're not allowed to ask questions. Emo, yeah.
That hybrid technique of the baiting and the RNA together.
Yes.
When do you think that the cost will come down for routinely use that?
I think you should go outside and ring up Treena. Sorry, I don't know. Yeah, I think, yeah, it's still a bit more time. Bill, any suggestions on cost? Yeah, the kit. The enzymes are really expensive, and the other problem is bulking up. You know, we're only using such small quantities of soil, and really what you need is bigger samples of soil. So the enzymes do large amounts of soil in one hit. That's going to be the challenge, but prices are coming down, hopefully, and techniques are going to keep improving. Yes.
And Giles, would you recommend it for the fallow trials that's going on?
The
fallow trial.
Well, that's basically what we now using these techniques here. We keep refining them depending on what we're finding, and so yeah. But I think both the DBCA and ourselves have been switched on with respect to using these techniques too. Bill.
Yeah, the sort of hybrid method, probably using the qPCR with baiting. You know, where you've got probably other pythiums and Phytophthora that overrun things pretty quickly. You know, you've got zoospores go to a bait, but you might have whole, you know, 2 or 3 orders of magnitude more pythiums, or to PC if it's in low, the inoculum load is very low, and then you might get, you know, one zoospore in 1,000 pythiums or other agar Phytophthora. So that's where probably the hybrid with the qPCA do a DNA extraction with the specific primer using the baits. Because if you play those out, it's probably just going to run away with other things and you miss it. That's probably where the strength.
And also
the hybrid method lies, where it's those really odd, low frequency
numbers
events that we're really interested in. We're probably going to pick up with the hybrid method.
And we also plate out leaves that don't show any lesions on as well. And again, and you can get recoveries from those as well. So yeah.
So part of the technique for areas where you're jointly symptomatic plants, so the tallow trials, for example, is a concentrating of the organic matter as well. So another step.
And
improving the process, but it's very time consuming.
Yeah.
And again, adds more expense.
Yeah. And that's a very important point, again, organic matter, because that's where Phytophthora is going to end up. You know, especially Cinnamomai is a poor saprophyte. It only colonizes living plant root material. Those roots die. That becomes organic matter. So when you sieve out as much of the organic matter as possible for your baiting and or your molecular tools, because that's where it's going to be. There's no point doing it from soil. Does that make sense? So again, with the fallow trials, etc., we're trying to get as much of the organic matter as possible, rather than just bulk soils. Yeah. So that increases your recoveries quite a lot. I didn't hear that. Didn't really hear the question. Are you using next generation sequencing? Yeah, we are using next generation sequencing as well, yeah. But yeah. I think I'm running into Sarah Navarro's type.
Yeah. Thank you, Giles.